Great. So, Marcia, thank you so much for the introduction. I want to welcome you all here today. Thanks for being here. I just want to kind of tell you ahead of time that I'm just a messenger here. All the work's been done by my postdoctoral fellow, Morteza Mahmoudi, and my two wonderful students, Michelle and Nicole, who are in the audience today. And I'm not going to repeat the title because it's too long. It'll be end of the talk by the time I finish saying it. So let's move on. So this is a cardiac MRI, and what it does to most of you, I hope you know, is that it provides a good contrast, a good image quality of your heart. And this is a female patient in my clinic who has long history of hypertension, diabetes, high cholesterol, who had 3-vessel heart disease and a bypass. She's New York Heart Association Class 2, meaning she could walk probably about 10, 15 meters without any problem, but we'll get shorter breath. And what you'll see over here is that the bottom half is moving, but the top half is hardly moving. In fact, it's going in the wrong direction. And when we do another imaging using MRI and look at how much of this heart is alive, this bright signal actually is bad. It means that the heart is dead. And you can see in this whole area, the whole thickness of the heart muscle is dead. And there's some what we call viability of live cardiomyocytes towards the end. And this is really a typical congestive heart failure patients that we see. And the question is what to do. And what we have been developing, and in fact this is what Nicole, one of her students, have done with Michelle's supervision, is to make these cells that we get from you beat again. And if you look throughout the bottom of the plate, you can see it's beating confluently throughout the tissue culture plate. And what I'm going to show you next is what these cells are actually secreting into the heart. And the question is it really these stem cells that's really necessary? Oops, sorry. Oh well. Is whether these stem cells are necessary or whether it's the secretion from the stem cells. And I'll go a little bit more into detail as we go on with the talk. So you'll see enough of those images as we move on. And so Joe alluded to this earlier, but there's a significant clinical importance in coronary artery disease and congestive heart failure. It's the #1 killer in the United States. I think we all know this. There's about 1 million new or recurrent heart attack, of which half of them result in eventual death. And in fact, all the diseases from the heart disease creates higher number of deaths in women than all the cancer causes combined. I applaud all the effort with all the cancer awareness that we see all the time, but please don't forget, coronary artery disease kills more than all the cancers combined. And thanks to some of the effort that Jennifer has been and her colleagues have been doing in the interventional cardiology, there's a shift in the landscape. So when you come into the emergency with acute heart attack, you actually survive most of the time. However, what happens next is that you develop heart failure. And heart failure is a #1 cause of hospital admission. In fact, it's a public health epidemic. There are about 5 million patients and about 300,000 new patients per year. And despite all the advanced therapeutics that we've been developing over the last 20 to 30 years, 5-year survival in patients with heart failure is a dismal 50%. Half of them are dead in 5 years. And of course, heart transplantation is a definitive therapy, but there are only 1,000 organs that are available for 5 million patients who need it. So clearly, there's a disconnect between what is necessary and what is available. And because of this discrepancy, the annual cost of these heart failure patients worldwide is over $100 billion. And what's more alarming is that female survivors of heart attack have a higher frequency of death, reinfarction, or congestive heart failure development than men. So by the time any female patients come in with any coronary artery disease and subsequent heart failure, we have to attend to it more seriously. And so there's a clear need for optimal therapeutic intervention for this critical unmet clinical need today. And so I was hoping that our proposal would address some of these needs in congestive heart failure and coronary artery disease. And these are the rationales for our proposal that we submitted. And I just wanted to kind of give you some introduction what we propose in our grant and what we have been able to generate. So clearly, male and female stem cells, Marcia have alluded to in her talk, they're clearly different. And male and female embryos differentiate at a very early developmental stage. And it is clear that there are genetic and structural sexual dimorphism prior to their hormonal changes as human beings develop into early teens. And in my mind, human placenta was the earliest source of stem cells, and it provided a wonderful model to investigate the basic effects of sex differences in stem cells. Before they're exposed to all these hormonal differences, these earlier stem cells must have some differences based on their sex. And that's what we're hoped to sort of investigate. So the objectives were twofold in this proposal. One was to look at the reprogramming yield of human placenta-derived stem cells. And these are all fancy words, and hopefully I can explain to you what I mean by reprogramming, placenta, and so forth. And we were looking at whether they could become cardiac cells and what type of immunoprivilege that these cells might have. In specific aim 2, then we wanted to see how these cells restore the injured myocardium, injured mouse heart in vivo. Okay, so what we have been doing is after the baby is born, we've been taking the placenta within like the first 30 minutes. And then what we try to do is isolate the amniotic membrane. An amniotic membrane is the interface between the mother and the baby. So it essentially protects the baby from the mom. If you think about it, delivery is a biological miracle, that this separate entity could survive within the mother's womb for that long before it gets attacked by the immune system. So what you have is an amniotic membrane that protects the fetus. And what we have discovered is that these amniotic membrane contain cells with very unique properties. First of all, they're very stem-like. Although they're already adult cells, we get it from the baby, they're very stem-like. They also have this special immunomodulatory properties, and more importantly, has this cardiac characteristics. And when you actually do gene profiling of these cells, they have a lot of pluripotent stem cell expression and also cardiac cell expression, suggesting that these are sort of pre-cardiac mesoderms, something you see fairly early on in embryonic development. And in fact, these amniotic membrane does come from the embryo early on in the embryonic development. So how do we isolate the amniotic membrane stem cells? So these are the placenta fresh out of delivery. We remove this outer membrane, which is called the chorionic membrane. And then underneath it, we carefully peel off this amniotic membrane. And this is a source of all our stem cells. Then what we do is then we wash and digest the membrane. And then next day, sure enough, you see this robust number of mesenchymal, amniotic membrane-derived mesenchymal stem cells. And per placenta, we get about 50 to about 150 million of these. And what we asked was a question. So what we found was that they're very stem-like. Can we make it really into a stem cell? Can we make it into pluripotent stem cells? And what we were able to do is that after treating them with what we call Sendai virus, which are Yamanaka factors, and I don't want to get too technical here, but these are genes that we treat these cells with. And by around day 10, they already look like they're pluripotent. And then you could see at least 20 colonies that we could generate from about 20,000 cells, meaning these guys are at least 100 times more efficient than skin cells in generating induced pluripotent stem cells. By day 20, they have a very typical characteristic of iPS colonies. And by day 30, these iPS, by passage 30, which is about 2 to 3 months after they become iPS cells, they become these contractile beating cardiomyocytes. And the fellows in our lab routinely are able to generate this very robustly from these cells. But it's both the cells and the genes that we treat them with that makes these cells, first of all, pluripotent, so embryo-like, And then we treat them with specific small molecules so they become these spontaneously contractile cardiomyocytes. And to this day, when I see the cells start beating by themselves, I get excited. And I've seen gazillions of these, but every single time you could feel that everybody is kind of excited looking at this. And so in a specific aim 1, the first aim was to see the reprogramming yield of male versus female amniotic membrane mesenchymal stem cells. And by reprogramming, it means that I want to make them pluripotent, okay? So these stem cells are stem cells, but they're not pluripotent yet. Pluripotent means it could become any part of your body. These mesenchymal stem cells could only become maybe muscles and bones and so forth. But we're going to take it back, all right? We're going to take it back, the development, and make it like right at the time that the sperm fertilizes an egg and they become pluripotent, they become any part of your body part. That's what we're doing, okay? And I hope that makes sense to all of you because that's kind of important. And that's what iPS cells mean. So initially, the male and female hMSCs, their stemness is quite similar. Now, once we reprogram them with these genes to make it pluripotent, it was really amazing to us that during initial state, the female cells were highly, highly pluripotent, whereas male cells were not. However, once they become iPS cells, they become pluripotent, and then into passage 10, which is about a month after we generate iPS cells, then the properties pretty much revert back. But the female cells still do retain the pluripotency a little bit more than the male. And this is what it looks like under the microscope. And this is just a sort of a serial pictures that we took in our lab comparing male and female at passage 20 of iPS cells and passage 30 of iPS cells, right around the time they were ready to become cardiomyocytes. And then there are a lot of genes that we could look at in terms of whether they're pluripotent or not, but these are some of the select genes that we looked at, and the names are not important, but we could certainly see that there's an increased expression in one of the genes in female cells and some in the male cells, essentially telling me that it's probably a wash at this point, that they have a fairly similar pluripotency capability. And then this is, again, the cells after they become iPS cells. We treat them for another, let's say, 10 days to 14 days with small molecules, and they become beating cells. This is a female counterpart, and this is a male counterpart, where, again, they're cardiomyocytes through the bottom of the tissue culture plate, and you can see them contract very nicely. And then what we're able to do is then to take it one step further and look at the physiology of these cells. And what we could measure is the contractile property and also the calcium signal, showing that the spontaneity, or the synchrony of the mechanical and the electrical motion of the cells. And unfortunately, this guy's not working, but this is a contractile cell that would glow in the dark because of the calcium flowing through the cells. And I apologize that this is not working. And so I was prepared. So this basically shows me that there's a calcium transient. You see this calcium flow with each contractile property. So they're happening synchronous. Again, demonstrating that these are really becoming early cardiac cells where their motion depend on the calcium flow into the cell membrane. And then you could then modulate these cells with beta agonist or antagonist. So isoproterenol is a beta agonist, so it activates the beta receptor. And although this looks like it's coming down, the heart rate is going up. This is kind of confusing, but the interval between the heartbeat is getting shorter and shorter, meaning it's going faster and faster. But if you treat it with beta blocker, then the heart rate is getting slower, or the interval is getting longer and longer. And then again, if you look at the contractility, the contractile force is going up if you treat it with beta agonist. And if you give it beta blocker, which slows down the heart and slows down the contractile property, the improvement is much attenuated here. And then we then do patch clamp analysis and look at a little bit more specific electrical properties of these cells called action potential. And we're able to show that there are 3 different types of these cells. One is a nodal-like, another one is atrial-like, but the majority are the ventricular-like cells, which are the beating cells. And then we're all working hard to be able to generate these type of cells, those type of cells, and those specific type of cells, but we're not there yet. At this point, we can still say that the majority are ventricular-like cardiomyocytes. However, all these cells are cardiomyocytes. In most of our tissue culture, we get a majority, 80 to 90% pure cardiomyocytes. Now let's move on to immunoprivilege. So what do I mean by immunoprivilege? Can I give my stem cells to you? Of course not. It's going to get rejected immediately. But what about these hMSC cells, which protected the baby from the mother? They are immunoprivileged. And so the whole question is, can I take these cells from the baby, make it into iPS cells? Do they still retain the immunomodulatory properties? So these are a lot of just stainings showing that these hMSCs have specific positive cell surface marker. HLA-G attenuates the T cell antigen presenting cells, essentially kind of recognizing the foreign cells, and also attenuates CD59, which is complement mediated cell lysis. Essentially, just a lot of fancy words for your own immune system going after other things, other foreign bodies. Well, hMSCs don't do that, and when you make them into iPS cells, you could see that they also retain those properties by having positive CD59, positive HLA-G, but negative HLA-DR. HLA-DR is a minor histocompatibility factor. Again, it just kind of allows you to recognize foreign body. These cells don't have that. So what does this mean? This means that maybe if I could take these cells, make it into cardiomyocytes, put it in the refrigerator, freeze it, and every time somebody comes in with a heart attack, we could inject those cells. It's a universal cell that I could give to anybody. And that is something that we hope to do in this field. Otherwise, actual clinical implementation of stem cells could be limited. And then if you look at the gene expression, again, hMSCs and MiPSCs are similar. And basically what this is showing is that the MiPSCs retain the immunomodulatory property of hMSCs. Okay, 5 minutes, so I'll try this quickly. Okay, so we injected these cells into immunocompetent mice, so human cells into immunocompetent mice. The regular human embryonic stem cells, they're dead. This is a signal for them being alive, but by day 2, they're dead. But when we took these MiPSCs and put it into mouse, regular immunocompetent mice, they actually survive up to day 10. And we also injected into the mice heart, and they were able to, although the signal is not wonderful, they survive at least week 1, and then most likely into week 2. And then here we have markers for different immunohistocompatibility factor. And what we saw here was kind of interesting. The female iPS cells had less immunogenicity, meaning that they were able to stay retained within a foreign organism better than the male. Okay, so the last section is, what about these cells restoring? The ultimate question is, all these basic biology is fantastic, but do they actually restore the injured heart? So we took all the hMSCs, the iPS, and all the different type of stem cells coming from the amniotic mesenchymal cells, and it showed that female cells perhaps, this is not definitive, might be better than the male cells in increasing the heart viability, allowing the stem cells to stay alive, and improving heart function. So what we were able to do is, using our sort of specialized contrast agent, show how much of the cell is, how much of the heart is alive, and what happens to this heart when we inject stem cell in this dead area. We were also able to use reporter gene to show that the stem cells are there and kicking and alive in the heart. And then finally, we wanted to look at the heart function. This is a mouse heart that got normal saline. This is a control. And then this is the one that got stem cells. And even for untrained eyes, you can see that this heart is contracting much more vigorously than this one. So using all these non-invasive imaging tools, we're able to assess how these cells are doing. So when we look at the EF, and this is still very preliminary, it shows that the female cells at least had a trend towards improved ejection fraction at 4 weeks. These are the hAMSCs, the iPS, and the cardiomyocytes. But what was more even interesting is that when we looked at how much of these stem cells are alive, it's really this teeny microscopic portion. That's it. That was all we saw were the cells that were alive that we injected. But we were able to confirm that these were human cells. They have both human antigen positivity, and then you colocalize in the area, and it all fits. But the whole idea is that it's a microscopic level of cells that are alive. However, if you look at the heart, all of this has come back. Those stem cells that resuscitate this much of the heart with just that microscopic remnant by week 4. And the question is why? And this is what biology is, right? This is just very complex, multifactorial. But these cells secrete all these factors called cytokines or paracrine factors. These are just good juice that's coming out of the stem cells, essentially. And then if you look at different cell types, they also secrete different goodies, different juice that's coming out of different stem cells. And what we decided to do was then not just to duplicate the different type of proteins that's coming out of the cells, but go a little bit higher upstream and look at what we call exosomes. And exosomes are these small balls that's being secreted from the stem cells that contain genetic instruction inside. Okay, and this all started in the oncology or in the cancer field, where there's one primary tumor cell that secretes what we call these exosomes and make the surrounding cells cancerous. Okay, and what we then were able to do with a lot of work from Michelle sitting in the back is taking these stem cells and showing that they secrete these good exosomes. And where we got them was just very simple. It was from the supernatant. We usually throw away these tissue culture fluid, but instead of throwing them away, we kept them and then we concentrated them. And here you can see these exosomes, and then these are what we call nanocytes. We collaborate with the folks at SLAC and using highly advanced tools to try to image them and analyze their size, look at the content, and then characterize in terms of what kind of microarray they contain. And I'll try to finish with this, is that we then injected the mice with these exosomes, and it showed robust restoration of the heart injury. This is normal saline, this is exosome, and the ejection fraction and the viability was similar to the stem cells. So this is what we kind of envision in the future, is that we take out the blood, we make it iPS, we differentiate in cardiomyocytes. Instead of injecting cardiomyocytes, which still has a lot of problems, we just take the exosomes, which is a cell-free therapy approach, and inject it back into the heart. And if you could kind of sit down and think about this, this is your own repair system, right? I'm taking your cells, taking the goodies out of your own cells, and injecting it back to them. Your body is constantly doing this to repair itself. We're just kind of giving a little boost. So we're hoping to use our Cardiovascular Cell Therapy Research Network, which is an NIH-funded network, of which Stanford is one of them, to conduct a lot of novel stem cell therapy clinical trials. And our hope is that by 2019, when this fund is going to expire, that we'll be able to use exosomes and then do our first clinical trial with them. So in conclusion, we were able to generate fundamental discovery that female hAMSCs and the derivatives have an enhanced effect on myocardial restoration through their immunomodulatory and anti-inflammatory effects. This led us to a future step where we want to look at the gender differences of exosomes. We clearly think that the female stem cells and their products have some anti-inflammatory component that would enhance the stem cell survival and myocardial repair to simulate endogenous repair of your organ. And then finally, I want to thank Marcia and Jennifer and all the donors for the high educational value that I was able to provide to our fellows and our students. And they learned this wonderful translation from basic science into clinical application. So thank you very much.